Section 55 of Gray's Anatomy, Part 4. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by David Lawrence. Anatomy of the Human Body, Part 4, by Henry Gray. 1d. The Organ of Hearing. Organon auditus. The ear. The ear, or organ of hearing, is divisible into three parts: the external ear, the middle ear, or tympanic cavity, and the internal ear, or labyrinth. The development of the ear. The first rudiment of the internal ear appears shortly after that of the eye, in the form of a patch of thickened ectoderm, the auditory plate, over the region of the hindbrain. The auditory plate becomes depressed and converted into the auditory pit. The mouth of the pit is then closed, and thus a shut sac, the auditory vesicle, is formed. From it the epithelial lining of the membranous labyrinth is derived. The vesicle becomes pear-shaped, and the neck of the flask is obliterated. From the vesicle certain diverticula are given off which form the various parts of the membranous labyrinth. One from the middle part forms the ductus and sanctus endolymphaticis. Another from the anterior end gradually elongates and, forming a tube coiled on itself, becomes a cochlear duct, the vestibular extremity of which is subsequently constricted to form the canalus reunions. Three others appear as disc-like evaginations on the surface of the vesicle. The central parts of the walls of the discs coalesce and disappear, while the peripheral portions persist to form the semicircular ducts. Of these, the superior is the first, and the lateral the last to be completed. The central part of the vesicle represents the membranous vestibule, and is subdivided by a constriction into a smaller ventral part, the saccule, and a large dorsal and posterior part, the utricle. This subdivision is effected by a fold which extends deeply into the proximal part of the ductus endolymphaticis, with the result that the utricle and saccule ultimately communicate with each other by means of a Y-shaped canal. The saccule opens into the cochlear duct through the canalus reunions, and the semicircular ducts communicate with the utricle. The mesodermal tissue surrounding the various parts of the epithelial labyrinth is converted into a cartilaginous ear capsule, and this is finally ossified to form the bony labyrinth. Between the cartilaginous capsule and the epithelial structures is a stratum of mesodermal tissue which is differentiated into three layers, viz., an outer, forming the periosteal lining of the bony labyrinth, an inner, in direct contact with the epithelial structures, and an intermediate, consisting of gelatinous tissue. By the absorption of this latter tissue, the periolymphatic spaces are developed. The modulus and osseous spiral lamina of the cochlea are not preformed in cartilage, but are ossified directly from connective tissue. The middle ear and auditory tube are developed from the first pharyngeal pouch. The entodermal lining of the dorsal end of this pouch is in contact with the ectoderm of the corresponding pharyngeal groove. By the extension of the mesoderm between these two layers, the tympanic membrane is formed. During the 6th or 7th month, the tympanic antrum appears as an upward and backward expansion of the tympanic cavity. With regard to the exact mode of development of the ossicles of the middle ear, there is some difference of opinion. The view generally held is that the malleus is developed from the proximal end of the mandibular (Menkel's) cartilage, the incus in the proximal end of the mandibular arch, and that the stapes is formed from the proximal end of the hyoid arch. The malleus, with the exception of its anterior process, is ossified from a single center which appears near the neck of the bone. The anterior process is ossified separately in membrane, and joins the main part of the bone about the 6th month of fetal life. The incus is ossified from one center, which appears in the upper part of its long crus, and ultimately extends into its lenticular process. The stapes first appears as a ring (annulus stapedius) encircling a small vessel, the stapedial artery, which subsequently undergoes atrophy. It is ossified from a single center which appears in its base. The external acoustic meatus is developed from the first bronchial groove. The lower part of this groove extends inward as a funnel-shaped tube, primary meatus, from which the cartilaginous portion and a small part of the roof of the osseous portion of the meatus are developed. From the lower part of the funnel-shaped tube an epithelial lamina extends downward and inward along the inferior wall of the primitive tympanic cavity. By the splitting of this lamina the inner part of the meatus, secondary meatus, is produced, while the inner portion of the lamina forms the cutaneous stratum of the tympanic membrane. The auricula or pinna is developed by the gradual differentiation of tubercles, which appear around the margin of the first bronchial groove. The rudiment of the acoustic nerve appears about the end of the third week as a group of ganglion cells closely applied to the cephalic edge of the auditory vesicle. Whether these cells are derived from the ectoderm adjoining the auditory vesicle, or have migrated from the wall of the neural tube, is as yet uncertain. The ganglion gradually split into two parts, the vestibular ganglion and the spiral ganglion. The peripheral branches of the vestibular ganglion pass in two divisions, the pars superior giving rami to the superior ampulla of the superior semicircular duct, to the lateral ampulla and to the utricle, and the pars inferior giving rami to the saccule and to the posterior ampulla. The proximal fibers of the vestibular ganglion form the vestibular nerve. The proximal fibers of the spiral ganglion form the cochlear nerve. End of section 55. Recorded by David Lawrence in Brampton, Ontario, September 2009.